Hey there, I just gave a brief presentation to some coworkers about the COVID virus and the mutational adaptation from animal to human, and I sketched it out on a whiteboard. Someone's asking me, Drake is asking me to do this again. So Drake, this is for you and your family, dedicated. So essentially, the coronavirus is called a coronavirus because corona is Latin for crown. It's got a bunch of spike proteins on the surface there. And if you look at the genetic code, about 76% of the genetic code for SARS version 2, which is what we have now, matches the old version right here. So this is old version SARS 1, it looks like. You can double check the data. The publications. I'll try to put some links at the bottom too. 24% of the DNA or RNA. I'm sorry, viruses are made of RNA, not DNA, but the RNA uses the same manufacturing, the parts inside of our cells to replicate itself. Anyway, we have RNA as well as humans. All animals, all species on this earth have RNA pretty much. Viruses are only comprised of RNA, not DNA. But anyway, so this part of it right here is new. It's sort of an upgrade. It's not actually this portion of the virus. But so anyway, the virus has evolved. The virus has changed. 24% of it now has shifted these spike proteins right here on the surface. These are actually proteins on the virus envelope. So this is SARS-2. Hold on, I gotta pause the TV. So essentially, these proteins on the virus have been upgraded from this little change over here, and they interact. I gotta be careful how I draw this. They interact with proteins inside of our lungs called ACE2. So ACE2 proteins are on cells inside of our lungs. And so you can think of, oh Lord, this is getting hilarious, huh? So these are little proteins that stick out in the cells inside of our lungs, typically in like the bottom lobes as well of the lungs. That's why there's kind of like that glassy looking structure or glassy effect on those x-rays. It's called fibrosis, and that's essentially scarring in the lungs. But the virus is seeking out ACE2 proteins. And once it finds it, it really binds on super tight. So this bind right here from the spike protein, the S protein on the virus envelope, it's a 10%, I think it's about 10x affinity. So it increased in its ability to bind super tight. That increases the infection rate, the efficacy of the viral envelope binding to our human cells. And so what happens once you breathe it in, it really grips on pretty tight again. So after that, you go into an inflammatory response. Now, that inflammation causes a cold or a cough, if you will. Essentially, it's like getting a splinter or cutting your finger. It's going to get red, right? There's blood flowing to it. There's white blood cells flowing to it because you're infected. You've got something that's foreign to your body inside of you. And this is airborne in spittle. So when people cough or sneeze, particulates from saliva can stay suspended in the air for about 2 hours before gravity pulls the particles down. If it touches a surface, it can live on the surface. The virus can live on a surface for anywhere between a couple hours to maybe 2 days, 2 or 3 days. Typically, that's not really typical from what I've read on coronaviruses. They usually last about 8 hours, maybe, if that. HIV, for instance, breaks down almost immediately as soon as it touches oxygen, touches the air. So different viruses have different structures on their envelopes that keep them alive longer at different temperatures and different humidity levels. So anyhow, so when the virus attaches to our lungs, what happens is you get an inflammatory response, and those are released by what's called cytokines. So cytokines are like communication material between cells. They're little signals. They're called peptides. They float in the blood. They traverse across membrane barriers. From the blood-brain barrier to your guts, all over your body, you've got cytokines just flowing between tissues. If a white blood cell or a tissue is traumatized, it can release those cytokines in the blood, and then your body will kick in an immune response, and then all the white blood cells that need to get there are going to hammer out that pathogen, that infection. So with COVID-19, some people are getting what's called a cytokine storm. And if you want to look up cytokines, it's cyto, meaning cell, and kine. I think it has to do with like enzymes or signaling, but maybe kinase. Anyway, so these signaling molecules are like super amplified when you get COVID-19 in some patients. That will cause a massive amount of respiratory distress. It'll cause the onset of is it ARDS, the acute respiratory disorder or syndrome. Essentially, it leads to suffocation of the patient. You can also get scarring and fibrosis in the tissue. Essentially, your body punches holes in your lung tissue, so it's the white blood cells are eating away at your own body. They're causing inflammation and devastation in your own body. That's the way like the Spanish flu back in the day killed so many people, because the cytokine level shot up so high, and that causes the temperature to rise up super high as well. When you get those cytokines, you go into like a septic shock essentially. Your entire body can start hemorrhaging. You can go into massive fever, and that's because this virus, on a microbiological scale, is able to convince our body that our body requires just a huge onslaught of white blood cells, and then your temperature will skyrocket, and it will stay high to the point you get organ failure and things of that sort. Also, the virus, the ACE2 protein, ACE2 exists ubiquitously throughout our body in a lot of different tissues. So there is a chance if the virus gets bloodborne, it could go and attack different organs as well. I think the liver has a lot of ACE2, and that can cause cirrhosis or liver shutdown, certain organs. I don't know if the kidneys also have it. It's rare. That's a super rare thing to happen because the virus stays in the respiratory tract. Also, some interesting things about the virus is that people will get like a dry cough or a dry sneeze, not too much of the upper respiratory area, but more of the lower respiratory area, again, the bottom lobes of the lungs. So the cytokine response varies human to human. You don't necessarily know if you're going to respond with a massive amount of temperature and inflammation. It's atypical. It's rare. With this virus, it's pretty rare. But it's obviously happening enough that it's caught everyone's attention, right? So it's something to be cognizant of and to look out for. But the whole reason why this part of the genetic code was able to modify itself and upgrade itself is it's because of the animal-to-animal interaction in, say, a live market. Animals are breathing the same air, and as one species of animal has a version of the coronavirus, because pretty much it's all over the planet. I think 40% of the common cold, there's a percentage of it, it's a lower percent, but it's caused by coronaviruses. Rhinoviruses cause a bigger portion of that. That's a different class of virus. But so the coronavirus, well, all viruses are modifying their genetic code through errors. So they make mistakes, but those mistakes are not necessarily bad mistakes, what's called deleterious, so like deleting. They actually can help optimize the bonding affinity of the virus proteins against our proteins. And once there's a match and they connect, it's sort of like Lego pieces. And they just click into place. So this is a much more significant with the COVID-19 syndrome, with the SARS-2 virus. Again, it's a tighter grasp on the ACE2 protein on our lungs. So as the virus was getting transmitted from one animal to the next, most of the times the virus would just die off. It wouldn't attach to the protein of that next animal species. But there was a chance. Now it's happened twice. SARS-1 and SARS-2. This is the second version, so there could be a SARS-3, on the horizon somewhere. But essentially, the virus was shape-shifting these proteins, these spike proteins, these attachment sites. The genetic code, it's a high variability region. I mean, it's designed to constantly change. even if it means bad things for that one virus copy, there's billions of other copies of that virus. So it's like, hey, it's like slot machines. You know, it just keeps pulling on that lever, and eventually you're going to get sevens all across. Once you hit that jackpot, meaning the pangolin or the bat or the camel or whatever animal is infected with that virus, that one virus, it hit the jackpot with its protein receptors that just so happen to match our proteins. So that means a human had to be there at the right time just when that enhancement occurred to then breathe in that virus. Because if there was no human there or no other animal to carry that one type of virus, it could die off. Or it could just keep replicating and create billions and billions of copies within that animal until somebody walks by or eats the animal or breathes the air or whatever happened. So again, look at the research out there. You can see how a lot of bats died from, I think it was the bat species specifically, had fluid in their lungs. Some researchers found that, and it was because of that, the bat version of coronavirus. And it took a while. It takes several years, sometimes centuries, for a flu or a rhinovirus or a coronavirus to, again, change a part of its genetic code in the high variability region to then manifest proteins on the envelope which match us. And then on top of that, you got to have a reaction that causes hemorrhaging, high fever, massive inflammation, and then acute respiratory syndromes, where the patient, again, will have just residual inflammation in the lungs to the point that the alveoli and what's called the interstitial tissue start to break down, start to get overwhelmed. And there's some publications coming out about patients, at least in China for now. We're waiting on the UK and the US to start producing all these papers as well. There's some younger patients who've left the hospital but have lost 20 to 30% of their lung capacity due to permanent scarring, maybe not forever permanent, but long-term scarring in the bottom portions of their lungs. So there's so much to be assessed and analyzed about this virus. A lot of it is known because of the first version of SARS. That genetic code, that RNA was sequenced. It was studied very, very in depth. A lot of not just the RNA, but the actual protein, the actual virus physically was studied as well. So now that the genetic code changed, it's sort of the blueprint, if you will, of the construction project there, meaning the virus being constructed. Well, now we're studying the new structure of COVID-19's virus, the SARS-2. So we're basing that off of the first model to see what was upgraded, what enhanced, what changed. And for now, it looks like specific portions of the spikes on the virus, again, are 10 times more able to contact our cells, or the ACE2 protein, at 10x affinity, meaning it's just 10 times more likely to grab on. Those numbers, all these numbers are subject to change based on computational models of what these proteins look like. There are some videos out there where people will walk you into the molecule through virtual reality. They'll show you the actual atomic structure of the protein. So data is cranking out on the research side. The NIH is an organization that matches up researchers, geneticists, bioinformaticians, those folks who work on the algorithm development, AI, machine learning, algorithms, all of that, and they marry that to the clinical side, the actual patients, right? Their symptoms, what they're dealing with, their antibodies, their cytokines, all the stuff in their body that's interacting with the virus. There are scientists on one side and physicians on the other, and organizations like the NIH are able to combine those data sets, and that's super powerful. And it's something that on a global scale usually doesn't occur this quickly. People tend to silo their data and keep it to themselves because they want to come up with a solution and a cure. You know, that's how pharmaceutical organizations work. R&D is very, very classified within the private sector. You don't want your competitor coming by and finding out what medicine you designed, because we can create medicine to attack the protein, the virus, or protect our protein so the virus can't see it. There's so many ways to mess with this virus. But right now we're sharing data. We're sharing it all across the world. We're sharing it very quickly. And that is because this is a pandemic and we all want to solve this. So in an unprecedented fashion, information is being thrown from one country to another through the cloud networks that we have in place. And the algorithms that we have in place are enhanced from the, how we structure them and design them to read viruses 5-10 years ago. We're upgrading our algorithms to hack this virus, reverse hack it as quickly as possible, determine what parts have kind of re-engineered themselves, and we can start hammering out medicines to help. The important thing to understand, though, is there are toxicity levels that occur within our bodies if we take the wrong amounts of even the right medicine, or if we take the wrong medicines, right? So just because a molecule destroys a virus doesn't mean it's not going to destroy us too. It could be a bad thing for our bodies. So it's really important that the FDA regulates and the WHO and all these other companies, CDC, they all have their eye on what drugs people are taking for what diseases, to ensure that the drug interaction with us doesn't hurt us. But if we can mess with the spike proteins on the virus, well, guess what? It can't see us anymore. So we breathe it in and there's no interaction because we'd have binding molecules that kind of clog up the receptors on the virus, and it won't be able to see us anymore or grab onto us. And there's other molecules that, there's so many ways. I won't get into it right now, but that's what's happening right now. There's the ability for the government to lift certain regulations and restrictions on physicians to give out medicines and try out new things that have been approved and that are known to be safe. So right now physicians are working with like drug cocktails, so combining Ebola medication, malaria, and HIV in different ratios and whatnot, and testing that out. There's a lot happening right now. The world is looking for a solution. So we understand the virus. We understand the virus's RNA transcript. We understand its genetic makeup. We understand how it's shifting itself to become a stronger, more virulent strain, a more effective particle in our bodies, an infectious agent. And we're going to find a way to stop it. It is a matter of time, and time is something that is really critical right now. So that's my spiel on what's happening. If you have any questions, feel free to post them. If I said anything wrong, post that as well. I will try to put on some publications on this video that I've been referencing and studying as well. So, and if you want to know my background, I used to sell genetic sequencing technology to scientists who would study RNA and DNA proteins as well, because proteins come on the other side of what RNA codes for. So you have DNA, RNA transcripts come off of that DNA through some machinery in the body, and then that RNA is transcribed into proteins, essentially amino acids that build proteins that build our bodies. So they're all tied together. It's a really elegant process. It's super complicated in a lot of ways, but then it's also super simple. It's a very interesting world to be a part of. And again, supercomputers right now are giving us an unprecedented speed at understanding dynamics at the molecular level. So now I'm getting super nerdy. Thanks for watching, and I hope this helps answer some questions. If it confused you more, I'm sorry. But anyway, I'll try to tone down the nerd factor a little bit in another video if you want. Thanks.